Thank you very much. It's a big honor to be here. I would like to thank Ole and Ditron for their kind invitation, and it's a big pleasure for me to be here and share with you some of my thoughts and ideas. Actually, the former speakers covered a lot from what I wanted to tell you, and there is very little information I'm going to add here in my introduction, but maybe I'm going to wrap it up slightly in a different angle to view all these subjects from a different perspective. Just wanted to make a disclosure. I'm the inventor and the founder of Nobio. That's an Israeli-based company that invented Kazi. And the first product is a dental composite. And we have a small stand which can give away some samples of this composite, whoever wants to try it in his office. Eventually, everything connects. An American designer, genius, Charles Ames. And he coined that the quality of connection is the key to the quality per se. And if there is something about the quality of connection in the dental area, Ditron manufactures the quality of connection. In the oral cavity, we have to really realize that everything leaks. And that's something that we have to really recognize the dentists, because this is maybe the phenomena that cause most, I would say, failures in dentistry, including some of the iatrogenic failures. And of course, we have this abutment implant connection, which leaks, and we spent quite a lot of time this morning already about this gap. So I'm going to add a little bit more information about this gap. Here is one example. So some very old implants you can recognize. And the same mouse, you see one implant is successful after more than 30 years. And the other one has bone resorption around it. And really the question is why the same mouse, same environment, one has bone resorption, the other one is successful, no bone resorption. So there is no one simple answer to this question, although we got so many aspects of this bone resorption, and we heard this morning so many hypotheses about the bone resorption, and this kind of situation does not really offer a good explanation why there is a big difference between these two implants in the same oral cavity. One of my students did this experiment many years ago, just to show that even recommended closing torque, you still have some leakage at the interface. So the point here I would like to stress that we should be very precise in closing our abutments in the appropriate recommended closing torque, because it is very significant about the leakage at the interface. Other aspect is the dynamics of the system. When it's in function and the forces of the abutment implant is not vertical, you get something that looks like this. And this is actually a type of a percolation. It's like a pump. And this results in what most clinicians experience in malodor when you reopen this implant abutment systems. And of course, one of the causes of peri-implantitis. And indeed, the abutment interface, you see an implant infiltrate inflammation at this interface. One of the subjects that was raised this morning is this inflammation and the tissue around this interface. What's very peculiar in the area of research in the recent years, understanding the inflammation, and more importantly, how the inflammation resolves itself. So there's something very interesting in the human body. It's easily turned on, the inflammation, but not every organism knows how to turn it off. And sometimes, even if you remove the cause, the etiology, you remove the bacteria that causes, or the macromolecules that are leaking from this interface, the inflammation is not resolved. So this is another aspect that you have to remember when we deal with peri-implantitis. As mentioned before, everything is contaminated, but the quality of the interface is really related to how much this is infiltrated with bacteria. And this is not new. 1993, bacteria were found in every single implant after 3 months in vivo. This is some EM pictures that I took with a student of mine, a screw that was removed for a novel BioCare implant. And you see beautiful colonies of this screw all over the place. So let me spend a few minutes about bacteria. Just to give you a small reminder, I spent 3 years of my life at the NIH nearby, working in microbiology. So in a way, I'm trained microbiologist as well. It's a small video just to give you an example. When we think about bacteria, we think about disease. And sickness, and pain, and immune response. So we work very hard to kill bacteria and to eliminate them from surfaces, from the body. But here is something very unusual about this. The first living organism on Earth was a bacteria. So just to give you an idea that this is part of our life, part of our health. When we say symbiosis, we mean that we coexist with bacteria. So just to give a little bit of proportion about time and scale, this I made this graph by myself based on David Attenborough. Probably familiar with him from the BBC. So if you take the 4.5 billion years of Earth, the first living organism, and you divide this scale to 12 months of the year, the first living organism, the bacterium, is about March. It took another maybe nearly 2 billion years to get the first eukaryotic cell. So they have about 2 billion years to create a diversity of living organisms on Earth. And even today, most of the living creatures on Earth are bacteria. Fish and plants appear in November, mammals on the beginning of December. December 26th, the dinosaurs extinction, and we humans appear 30 minutes before midnight. And you cannot avoid the conclusion that we evolved from bacteria and we evolved among them. So there is a big chunk of evidence today that every single living multicellular organism on Earth has its own specific microflora. Ants, cockroaches, snakes, monkeys, and ourselves. Each of these creatures and many more have its distinct microflora, very specific for them. So the coexistence between bacteria and us, humans, is a very fundamental feature of our life, and it's necessary to keep our health, not disease. So with this basic understanding, we also have to recognize that 37 of our genes, 37% of the human genome have homologous genes with bacteria. And another 28% come from single cell eukaryotic cells. So the symbiosis has a different meaning to just coexistence. It is part of our well-being, part of our health. The other thing that is recognized in the last 20 years in the world of microbiology, that bacteria does not exist floating in the air or swimming in the water or in saliva. The way bacteria exist on Earth is in form of biofilm. And biofilm is a multi-species, multicellular organism. Actually, you can view biofilm as a multicellular, most primitive organism. And you all know that they are very resistant to antibacterials, antibiotics, and to the immune system. When you say resistant to the immune system, we don't really mean that the immune system fights bacteria. The immune system is trained by bacteria, and most of our bacterial genome associated with the human genome are good bacteria, maintaining our health. So not necessarily dental plaque bacteria are bad. In killing all bacteria or killing all the plaque bacteria, you're not doing a good thing to the human body. Actually, you may complicate things. So what's known today about dental biofilm? 2,000 different taxa. Only less than 50% is culturable in the laboratory. You cannot culture them or grow them. This data is coming from next generation sequencing data. Every single moment, each of you in this room has between 600, 700 species. And one is different from the other. The diversity of all species of a human being is more accurate than a fingerprint, very specific to a specific person. And there's a lot of information around it. I don't want to spend the time about that, but I would like just to point out that only 100 from all these 2,000 different taxa are associated with gingivitis and periodontitis. When we say associated, that means not all of them are causing disease, but when you have an association with disease, you have an increased number of this in the samples from the disease. And we are not already talking about the era when P. gingivalis or AA are the most important bacteria for gingivitis or periodontal disease. And we are not talking about having 10 different caries-associated bacteria. And strep mutans is not the leading one anymore. It's a specific interaction between several different types of bacteria in a specific site that causes the damage, that turns on the inflammatory reaction that in some patient cannot be turned off. So it's very simple to stand on the podium and tell you its life is very simple, but probably it is much, much more complex than we can imagine today. And I was talking about 2,000 taxa of bacteria. In each taxa, there are several hundreds or maybe thousands of species. And the diversity is really enormous. Just to give you some examples, to remember, many patients have P. gingivalis and AA, have no periodontal disease. Many patients, and we know it for sure today, have lots of strep mutans and strep salivarius and mitis, no caries. It's not enough to have the bacterium to have the disease. These are really recent publications in the last 5, 7 years. So let's go back to this case. I think it's pretty clear to everybody that we have a gap, and we have also a specific microbiome associated with this gap. I've already demonstrated in so many papers that this is harboring bacteria. There's a species diversity, and it's also a polymicrobial biogeography. There are some papers out there to show you a very specific three-dimensional structures associated with it. Structures, I mean, microbiological structures in the dental plaque. So just imagine that all these spaces in the red dots are covered with a biofilm, which is extremely difficult to remove. Once the bacteria entered in this space, around the screw, in the basket, along the implant, along the abutment, they're extremely difficult to remove. Now, if these bacteria are good ones in association, you won't get any disease. But if these bacteria are bad, potentially pathogenic, turn on inflammation, you have a real problem. And I'm looking at the literature about treating protocols of peri-implantitis. Very few are mentioning to take out the screw and to sterilize the internal parts of the prosthetics, the abutment. As somebody mentioned before me about it's very similar to an endodontic treatment, but not because of the bacteria that penetrates through the endodontic canal to the apex. It's because the dentin is invaded with bacteria, and these bacteria are going to the side to the PDL and cause the inflammation. And you cannot get rid of these bacteria in some endodontic cases. Same here. You have to take all the parts apart in order to cope with it. So just imagine a situation when you can produce these parts, these titanium parts, being covered with some kind of a material which is antibacterial. So in a way, you are lowering the risk of having this kind of reaction, host reaction of inflammation, to turn on the inflammation process. So if I have to make a guess from what I understand in microbiology, the difference between these two implants is the type of microorganisms that harbor all the internal parts of this element. And yes, the problem is also in orthopedics, not only in dental implants. And in a way, we learn from them and they learned a lot from us. And in a paper that published recently, they are talking about anti-infective coating of implants. So that applies also to dentistry. They are looking for something that is not there yet. So here is our invention. It's a particle, nano-sized particle, covalently binding quaternary ammonium, and that makes the whole particle and the surface that is embedding the particles to be very potent, broad spectrum bactericidal surface. A quick example, a surface control, bacteria, 1 million bacteria on the surface, that's SEM pictures. 24 hours later, you see billions of bacteria, biofilm. Same surface harboring 1.5% of the technology. After 1 hour, you can see some bacteria exploding, cell membrane damaged. 24 hours later, no one single living bacteria. If you take a Petri dish, you put a lawn of bacteria, you place an antibiotic disc, you get a halo of inhibition. You put samples with or without particles, you don't see any halo of inhibition. Nothing is secreted to the surrounding. Nothing is secreted to the surrounding. So either no bactericidal effect or the antibacterial effect is non-soluble. As you saw before, is a very potent antibacterial effect by contact only. This experiment was done 6 months of aging in PBS. This is normal bacterial growth on the control, and on the Nobio containing, the CASI containing 1% of the particles, no bacterial growth after 6 months. So it's not only non-leaching, but because it's not leaching, it's very long-lasting. And that's the first antibacterial composite approved by FDA. And they allowed the claim for the first time for a dental restoration that adding CASI particles reduces demineralization. It's part of the caries forming process. Let me just run through it. So we did it also in coating titanium. So it's a normal titanium disc. That's a cooperation between Nobio and Ditron. They supplied us the titanium discs. They were coated with Nobio particles. You place on the disc bacteria, then you stamp it on a Petri dish, an agar. You remove them, and that's what you get. No bacterial growth on the Nobio coated surface. In a different experiment, what we did, we placed bacteria on the discs, and we see how many bacteria can be removed or recovered from that. And this is the bacteria recovered from the titanium control. And the Nobio particle-coated titanium, no bacteria could be recovered. Control, cytotoxicity and biocompatibility looks very much like the antibacterial coated abutment. No difference. Histologically, 6 weeks post-surgery in dogs, no visible differences between. So the potential is there. And what we believe is happening is the following. The peri-implant host microbiome balance. And this balance is dancing back and forth between symbiosis and dysbiosis. And our fight is to push it towards symbiosis. When you think about it, it is amazing that we are placing implants in the most contaminated part of the human body, and it's successful. If somebody would tell you this 50 years ago, we would say you are crazy. It will never work. But somehow, the good bacteria is helping us, even here. So if we are talking about dysbiosis, we get peri-implantitis, metallosis, bone resorption, aesthetic problems affecting adjacent teeth and implants. And we have a very good evidence in the literature today that by improving hygiene and maintenance, you can push all the system back to symbiosis. And with that new technology, this is additional layer we can add to our protection of the dysbiosis, is having an antibacterial titanium implant. Thank you very much.